Hello and welcome to this webinar titled How to Utilize Design of Experiments, Principles for the Development of High-Throughput, Robust Methods for the Assessment of Product Quality, hosted by Biopharma Asia magazine and presented by Jeremy Springall, PhD, Scientist 2, Analytical Sciences, R&D, Biopharmaceutical Development, AstraZeneca. My name is Stephen Edwards and I'll be your moderator. Before we begin, I would just like to inform our viewers that there will be a live question and answer session after the presentation near the end of the webinar. Audience members can send their questions in at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now please allow me to introduce our presenter. Jeremy Springall has worked in the Analytical Sciences Group, part of R&D Biopharmaceutical Development at AstraZeneca for the past 5 years. His responsibilities include assessment of new technologies and work processes to support early and late-stage development assets, as well as being a CMC analytical team lead on several non-MAB projects currently in the AstraZeneca development pipeline. Previous roles include the in-process analytical development scientist at UCB and analytical development scientist at Patheon, both in the UK. He holds a PhD in bioorganic and medicinal chemistry and a BSc in chemistry from the University of Bath, UK. I will now be handing over to our presenter. Welcome, Dr. Springall.
Thank you, Stephen, for the very kind introduction and for also inviting me to present what I consider to be a very exciting topic. So my talk today is going to be on how to utilize DOE principles for the development of high-throughput methods. And it kind of talks through a case study with then an application on process analytical technologies, or PAT, as we get through the slides. And this is something which we have implemented and are proceeding with within AstraZeneca. So as an overview of the talk, really, why do we need high throughput methods? You know, why can't we just do what we've always been doing? Why do we want to use the DOE principles for the development of these kind of methods? And then there's an overview of how we performed an assessment of comparability between the new method and the current platform method, with then a summary overall towards the end. So why do we need high throughput methods? Well, biopharmaceutical development can be exceedingly challenging, as most people I'm sure on the line know. It's a highly competitive environment with a lot of really incredible science that goes on. There is, over really the last 10 years, there's been such an increase in molecular format complexity from the traditional monoclonal antibodies, so MABs. We now have things like antibody-drug conjugates, bispecific, like non-covalent fusion structures. There's a whole heap of engineering colleagues seem to have an endless imagination as to the formats that they can construct. So this really changes some of our business processes, because typically when we were focusing only on MABs, we basically had a really solid platform where we could just push molecules through the pipeline. But now with these new formats, we need to think about how we should do business in a slightly different way. There's also a change in business processes around how we can do more analytics with less. And accelerated timelines are always something that is a challenge for our industry. So it's how can we get to market faster without impacting product quality and efficacy, because we know there is a patient need for these new medicines, and that we want to be able to ensure that we can help people lead healthier lives. So with all of this, you know, there's a lot we need to do to be able to meet all of these challenges. And really, one of the biggest ones is to be able to make sure we can do our testing faster. So high throughput, there's a lot of high throughput process development technologies out there, particularly in the manufacturing environment. So when we consider upstream, we go from our classical 3-liter bench scale bioreactor systems to these very, very high throughput, 20 mil kind of systems. So we have automatically increased our throughput by 24 to 48 fold with a lot less volume than what we historically did. And in terms of downstream, where we have our classic actor systems, we can now perform a variety of purifications, using robotics. So the examples shown here kind of show that from upstream we can increase our throughput by 24-fold, and through downstream potentially by 8-fold, if not even higher, with new technologies that are becoming more available on the market as we progress through. So with this, size exclusion is really one of the biggest tests that we perform to support our process colleagues. And when we think about it, in cell culture or upstream, we might have a cell line development campaign where we might have 96 samples per campaign, and we might have multiple campaigns going on at any one time. For clone selection, out of the cell line development, we could have 24 to 48 samples per day for roughly 6 days of campaign. So the numbers are starting to multiply quite quickly. And when we consider bioreactor optimization as part of later upstream characterization and process optimization, again, this number can range dramatically depending on how they construct their study, whether or not they utilize DOE principles as well. And then for protein purification, we can do resin screening really easy using robotics, and this can be up to like nearly 400 samples a day. Using scale down models, we can get nearly 50 samples a day. And then again, bench scale gradient purifications could be up to 20 samples per experiment, and this could typically take place over 1 to 2 days as well. So when we consider all of this, our current size exclusion method that we have it has a 20-minute runtime. But if we think about the number of samples we will see per day from upstream, it could be 144 samples, which equates to a total runtime of maybe 3,000 minutes or 2 days in total. For purification, this could be 452 samples a day, which equates to roughly 6 days. So when you think about it in terms of the amount of time it takes them to generate a sample, they could be waiting just based on runtime up to 8 days as a fastest turnaround just to get that data back, which really puts Analytical in the crosshairs for how we can accelerate projects and facilitate our development colleagues effectively. So what do we need this method to do? Well, really we approach this from the kind of analytical quality by design approach, or AQBD, or sometimes described as the enhanced approach, where we wrote out an ATP, so an analytical target profile up front, and this really defines the objective and the quality requirements of the reportable result, and it's also a prospective summary of the required characteristics as well, and it's a way of ensuring the data is fit for purpose. For our definition of this for this method, it was that the method must be able to determine the relative quantity of major product peak and high molecular weight species in drug substance, drug product, and in process samples. So the method must be specific, it must have accuracy and precision. We need to detail the sample handling so that the sample handling required does not negate any time savings due to faster runtimes. And really for us, the speed of the method, it must result in a reduction of runtime by at least 2-fold. So we've included a couple of non-classical examples of kind of requirements for the method around the sample handling and speed of the method. But for us, this really facilitated deciding when method development was finished so that we could progress with implementation in our development environment. So why do we use DOE for this? So this is really just a general model for DOE, and described by Douglas Montgomery in his Introduction to Statistical Quality Control. DOE is a series of tests in which purposeful changes are made to the input variables so that we may observe and identify the corresponding changes in the output response. So in short, we have a number of input factors. This could be things like mobile phase pH, flow rate, UV detector wavelength. We have some controllable variables that we have. So this could be, for example, how we trained our analysts to make up the mobile phase, because so it's a kind of controlled variation. Uncontrolled variables could be the laboratory environment. And then the output responses are kind of the quality characteristics of the reportable results. So for us, this was things like resolution between the high molecular weight species and the major product peak. It was number of peaks detected. It was runtime, things like that, for example. And we were able to use the DOE to evaluate the effects of the most influential parameters upon our reportable result, identify these interactions, and then also optimize to our best operating conditions. So, classically people have used one factor at a time, or OFAT, to basically develop methods. And this involves varying one factor at a time to provide estimates of the effects at set conditions. So this is on the left-hand side here, the box there. So you might optimize to the x-axis first, then to the z-axis, and then up on the y-axis to get your final conditions in green. When we look at DOE, so when we consider, for instance, a fractional factorial optimization DOE or a full factorial design on the right-hand side, we actually gain a lot more information around the entire space within the cube that you don't get from OFAT. So one factor at a time really finds a local optimum, but DOE can find a robust optimum. And through adopting the DOE approach, we ensure a thorough assessment of risk, and also it enables us to generate an appropriate mitigation strategy if we need to as well. So really, how do we look at this for design of experiment for robust analytical methods? Well, we use it to assess the design space and any potential factor interactions really in a stepwise approach. So we started with a screening. So this is a classical kind of Plackett-Berman model design that you might use. And based on the output from that, we identified the most statistically relevant factors for separation performance. We then took these into an optimization DOE. And then furthermore, once we identify what we consider to be a robust range, we performed a robustness DOE, which is in the darker purple square in the middle. So your optimization DOE kind of covers, your factors would be at typically like 2 to 3 times your SOP control range. So, and then your robustness is at your SOP control range. For us, it really led to an increased method knowledge. So we used this approach where we decided to use the screening for looking at a number of UPLC columns. So we looked at 11 columns through this initial assessment. Based on this, we then decided to do an optimization off the top 2 columns where we looked at assessing the impact of flow rate, mobile phase, ionic strength, and pH. And then when we identified our lead conditions with lead column, we assessed the impact of minor changes of these variables on the method performance. So we ended up with what we considered to be a really good separation of a very complex sample. So on the top chromatogram, we have the current platform SEC method. So this is the 20-minute method that we currently run. You can see that there are 3 very distinct high molecular weight species peaks and then the major product peak there. Then when we look at the bottom chromatogram, this is the 5-minute method that we ended up finalizing on. You can see that there is a minor shift in resolution between the earliest eluting high molecular weight species and the middle one. However, we felt that this was acceptable because we report total high molecular weight species, and the resolution between the front shoulder off the major product peak was comparable to that of our platform method. So for us, we felt that this was really good. We ended up with a runtime that was 4-fold higher than our previous method, so exceeded our expectations from the ATP. And the species that observed were visually comparable from their profiles, and that the percentage areas were also comparable when we performed the integrations and reporting of this as well. So we have a method, great. So now what do we do with this? So size exclusion is really typically a platform method that's used across multiple projects within a company's portfolio. So we really now wanted to assess that now we have this one method, can we apply it to other formats and sample types to truly make it platform like our longer runtime method is? So we decided to perform a really in-depth statistical assessment of the comparability between these methods. And so we ran a lot of samples. We basically ran across, I think it's about 6 different molecular formats with high molecular weight species ranging from less than 1% up to 30%. And we then pulled all of this data to perform the analysis, but we really tried to push what our understanding was of the method and how we determine comparability. We decided to do a couple of regressions, and what we see on the left-hand side here is the regression of the major product species, where basically what we have plotted is the high-throughput method on the x-axis, and on the y-axis is the low-throughput method, so the platform method here. And we did a regression between the two. So the line of equivalence, so if the values were perfectly aligned between the 2 methods, is the green line, is the green regression shown in the plots. What we have is the red line is the regression between our actual data sets, and it's a bit small, but we also have a 95% confidence interval of that fitted line shown in pink as well. And what we can see is that actually we have very good regression between the 2 methods. However, there appears to be a proportional bias between them. So as we get to increasing levels of high molecular weight species, the variation between the 2 methods increases. So we have a much better closer agreement at lower values of high molecular weight species than we do at higher values. And our confidence interval for the fitted line does not contain the line of equality. which is the if they were equal. However, our RMSE, so our root mean squared of the high molecular weight species on the right-hand side, is not bad, 0.96. We feel that was all right, considering we analyzed 263 samples for this assessment. So when we did a bit more of an in-depth inspection on the data, we were able to identify that there appeared to be some differences based on the molecular format. And this really had the biggest impact on the variation seen between the two methods. So what we did here, we plotted the relative percentage difference between the two methods on the y-axis and then the formats on the x-axis. And what we can see is that there are some which have very, very good agreement. So for instance, format 1 and 5 have very close agreement. Format 3 and 6 tend to vary a little bit more than what we were considering. So this really led us to do a bit more of an investigation into the root cause, and we found that a possible reason for this was that the differences were identified as being due to sample solution stability. So there was a really key learning from us to take this onwards that I think is useful for everybody when you're designing these kind of studies, is that you need to ensure you have adequate control for your sample handling to ensure you generate accurate comparisons of your data sets. So for us in this example, we found that we might have run on the platform method, say 2 to 4 weeks prior to running on the high throughput method, just due to resource capabilities, and that the sample was stored in a 2-to-8 refrigerator. However, during this time, there was aggregation that took place in the vial during the storage. So for us, we've now implemented a way of doing this where if we're unable to run it within 2 days between the two methods, we now freeze the samples within a -80°C freezer, and then we know that we have integrity of the sample between the two methods. We decided then, because we saw this kind of variation in the regression, to look at a limits of agreement determination. And LOA really is the maximum allowable difference between the methods for which they are considered to be in agreement. So there's a couple of ways of doing this, and one of the classical ways is the standard Bland-Altman approach. And really this is where your mean and standard deviation are constant across the range and your results are normally distributed. However, for us, this isn't necessarily the case from our data set. We tend to have a slight non-normal distribution, and our mean and standard deviation is not consistent across the range we're looking at. So we have utilized a modified Bland-Altman approach, which can be seen in this reference here from 2003. And from this, we're able to determine the LOA for the new high-throughput method. So when we look at these plots, they kind of, they need a little bit of describing. So what we have is on the left-hand side, the major product peak, and the right-hand side, the high molecular weight species. What we have is our regression from the original plots as through the middle of it, and these kind of like ever-expanding dotted lines that take place is the range in which the methods are considered to be in agreement. So it's like a statistical agreement between the two methods. So for instance, if we take 20% of the high molecular weight species on the right-hand side from the HT method, this is equivalent to 17 to 26% on the platform method. So this is more of a statistical ranging. However, this 20% is actually a very high percentage value of high molecular weight species, but typically we would see much lower than this. And this is why you can see such a grouping at the lower end between 0 and 5% of the high molecular weight species, where the majority of our samples are. And with that, our LOA at those levels is much, much tighter as well. Now, the two top plots at the top are if we lumped all the data in together. When we decided to split the data out by molecular format, in the Bland-Altman plots now shown at the bottom here, we can actually see that there is a much tighter LOA agreement if we consider them all individually. So what we have is, these are the classical plots where your mean difference is 0 on the y-axis, and then you can see your actual mean value is the solid red line going horizontally from left to right, and your confidence interval is then the dotted lines going left to right as well. And so really for the methods to be considered statistically equivalent, your 95 confidence interval must span over 0 mean difference on your y-axis. So for a couple of formats we have this, but for others we're just a little bit off. And this is relative percentage difference, not absolute percentage difference as well. So from this, we got a really great understanding of the capability of our method based on sample, molecular format, etc. And this really, we felt that when we presented this and we talked about this internally, that this presented an opportunity for implementation of process analytical technologies. And we got agreement from our purification and other manufacturing colleagues that actually this was a suitable method for us to proceed with. So we decided to do a number of proof of concepts to be able to support this as well. So when we consider PAT, this is kind of all about, in my mind at least, how can we ensure that we either acquire data to monitor our manufacturing processes in better ways, or how can we use the data we generate during this to control our manufacturing processes to deliver a product quality that we desire. And for us, high molecular weight species is a very, very key product quality attribute, because there's a lot of literature evidence that suggests that high molecular weight species can lead to immunogenicity and other undesirable. So really, we were thinking about this in terms of how can we put this in line to potentially control a purification step where we want to cut out during the purification column elution high molecular weight species. So we used an instrument to be able to do this. We started with our capture affinity elution product. And what we can see on the right-hand side here is an example of the viral inactivation where we did this step over. So in classical biologics manufacturing, you need to have 2 viral inactivation steps that take place in your manufacturing process. And really the first one in most examples, if you have an affinity capture step as your first purification step, you tend to elute off that at a slightly more acidic pH. So you would then lower your pH a little bit further for a period of time, which you determine to be inactivating to the virus and then reneutralized to progress on with manufacturing. So what we have here is a very, very small scale proof of concept where we used a beaker on a stir plate for the viral inactivation. We then use the instrument to draw samples to run on the instrument to measure the high molecular weight species. So this performs an online dilution prior to and neutralization prior to injection to maintain the integrity of your UPLC column. So what we were able to do with this was we were able to show that the aggregation rate is comparable when using the PAT instrumentation for the scale-down model to our large-scale characterization data. And when I say large-scale, I'm talking over 500 liter within this. So what you can see from the graph below is that there is a slight offset in the starting levels of high molecular weight species, and this is really just due to the starting level of the sample that we performed the study on. It's not due to the performance of the step itself. What we're really looking at is for comparability between the slopes, and we can see that from this here. So our actual rate is predicted in blue from the instrumentation, whilst our model predicted rate from our large scale is predicted in red, and we actually got very close agreement with this. So we feel that there is a lot of potential for this kind of technology to be used within a manufacturing environment, and it does require some vendor interaction to kind of make sure that the quality systems and stuff are there, and the vendors are very supportive of this. But really, we do see this more broadly implemented across a variety of our platforms now and manufacturing facilities at the same time, and across not just the size exclusion method. We are actually looking to implement this across a variety of different high throughput methods that we've been working on over the last 24 months. And we're in the process now of putting these in the manufacturing environment at the same time. So in summary, we really have successfully developed and implemented a new high throughput size exclusion method that exceeded our expectations from the analytical target profile. So it's actually 4 times faster as opposed to 2-fold faster. We have implemented this in cell line development screening, purification development screening, formulation as well. And herein I've described an example of a PAT application, but there are many more of which we're implementing now. We are working with other departments such as quality for determining a path forward for more broad implementation, and also with our manufacturing colleagues to ensure that we are listening to our customers to make sure that we can deliver to expectations as well. So some of the key learnings that we took from this are sample storage is very, very important. It can be easily overlooked, as I've described herein, but it's something where when we generate these comparability plans now, we have to ensure that we communicate the sample storage requirements to anyone who is generating the samples and the analyst who is performing the comparison. It is also very important to perform your comparison on samples that you are likely to see upon implementation. So one of the first things we did was we took some very, very early research constructs, which we would never see in our kind of development portfolio, and we ran it on there, and we saw some differences that we didn't expect, and this was really due to them being non-optimized protein constructs and non-optimized buffers, again leading to in-vial aggregation due to poor solution stability and also poor protein stability in itself. But when we did the reassessment using development material across a number of our more traditionally optimized processes, the comparison came out much, much, much closer. So it also means that we've understood what environments we can implement this method into and ones which we can't at the same time. Yeah. So in terms of acknowledgements, as you can imagine, there's more than just me who's been working on this. We have a number of colleagues who are both still within AstraZeneca and have moved on to other opportunities from physicoanalytical sciences. So this would be Sophia Levitskaya, Max Avansky, Eric Menke, my line manager, Wei Zhu. We also have colleagues in our bioprocess technologies and engineering group. So Lindsay Arnold has definitely been a great collaborator of ours for the assessment of these technologies in what we would consider to be a live environment. And then from our protein purification sciences group as well, Caroline Kittinger and Adrian Mann. Again, can only say thank you so much for generating all of these, well, the majority of these samples to us. And then from data sciences and modeling, James Savory and Kelsey Newell for really supporting the vision of the comparability assessment from the statistical approaches to then helping with the communication to a broader environment within AstraZeneca for why we feel this is the best way to perform these assessments moving forwards. And really with that, I would just like to say thank you for your time and for listening. And please feel free to send any questions through. And again, thank you, Stephen, for inviting me to perform this presentation.
Thank you for that presentation, Dr. Springall. Now, we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now, our first question is, how do you decide on what DOE design to use for the method development?
Yeah, this is an interesting question which we kind of wrangled over quite a bit because, as you can imagine, there's a number of DOEs that are available for this. So the way that we approached it was we wanted to do this in a stepwise manner to try and minimize the number of experiments we were running. Because we thought there's no point in doing a DOE if it's going to take much, much, much longer than the classical one factor at a time. So we really decided to take a very stepwise approach to this. And so we proceeded with the Plackett-Bowman screening design, first of all. And based on the outcome from that, we were able to identify the most statistically significant parameters based on the minimum number of experiments we could do. And from that, we then did the fractional factorial design with 3 center points to understand how that variation across the design space took place to identify potential robust conditions. And with this, we actually found that there were a number of secondary interactions that took place primarily between mobile phase, ionic strength, and pH for separation performance. And from there, we were able to elucidate out what we consider to be robust conditions based on our risk assessment of it and our risk mitigation, our level of risk that we were acceptable to have. And then we performed a robustness DOE, which basically was at our SOP condition. So for instance, if your target mobile phase pH is pH 6.0, we performed our optimization DOE from 5.7 to 6.3, and then our robustness DOE was 5.9 to 6.1. So we could understand really a broader variation across there, but then the variation within our anticipated SOP conditions, which would have been pH 6.0 plus or minus 0.1. From this as well, by gathering all the information from the optimization DOE, if we saw something that kind of came back that was very unexpected, and we determined it not to be due to the sample, we could really use that historical DOE data to drive an investigation to identify the root cause of that variation. So for us, we feel that there's a number of benefits to utilizing DOE for this, and following the stepwise approach minimizes the number of experiments you perform, but also maximizes the data that you get out at the same time. So I would say, yeah, screening, optimization, robustness, using a placket Thurman first of all, followed by fractional factorial central composite designs is how we approach this.
Thank you. Our next question is, how do you think this fits into the concept of analytical quality by design?
So the AQBD approach is kind of becoming more and more of a hot topic. There's been a number of workshops, both from the USP and more broadly at a number of conferences on this. And for me, DOE is integral to the approach. So really the whole QBD approach is that you theorize and define, well, not theorize, you define what your intended method purpose is up front, so that you know whether or not you've met the requirements of it. And it's a really useful tool for defining when method development is complete. So you don't just keep tweaking unnecessarily. And really from that, based on how we did it here, where we defined our ATP up front, our analytical target profile, we then were able to utilize the DOE to statistically say, yes, we have met the ATP. And based off that, we then were able to describe what we would call a method operable design region, or MODR. And from this, we then were able to progress very quickly into what we would consider to be a suitable method qualification or validation strategy for this, where we're able to leverage historical data to assist with that as well. And then based on that, we have now implemented a variety of continuous method performance monitoring processes, which enables us to ensure the method is within its desired method quality performance characteristics, so that we can then trust the data more in depth than ever before. And so for me, the DOE is really integral to the AQBD approach with that.
Our next question. In terms of implementation of the method, what is the current state and your future plans?
So the current state is that the method is now implemented and up and running, which is great. We have it across a number of labs and across a number of countries for which our organization is situated. In terms of future plans, really, ideally, if the opportunity arose, and it has to be driven really by the project opportunity, not by our entire desire, is that we would love to have this sitting in a GMP manufacturing environment to assist with process control, so that we can look to acquire more data to understand our manufacturing processes in greater depth. And really, one of the things that I see these high-throughput analytical technologies really assisting with in terms of PAT, so the process analytical tech, is that when we are able to acquire more and more data faster and faster, that we are able to utilize it, we can then really gain an in-depth understanding of what we determine to be our quality profile of our product from a manufacturing variable perspective. And when I say manufacturing variable, I mean, say I take my capture affinity step. There's a lot of processes that take place within that. It's often overlooked in terms of how complex those steps are. So you've got resin to resin variability. You have like purified to purified variability, meaning that your flow rate can be slightly variable between the two. You've got someone making up a buffer versus person B making up a buffer, so there's variability there. So as soon as we can start to capture all of this information in a better way, we can then utilize these larger and larger data sets to be able to really make accurate and highly predictive in silico models of our purification, which then means that in future our process development could probably be very, very much scaled back, because we have an exactly high understanding of the process performance due to data that's been acquired more rapidly than ever before.
Thank you. Next question is, what is exactly the relation between PAT and DOE?
So for me, they are quite separate. I think there's a couple of ways to look at this. So if we think about like PAT in terms of methodology, so just the actual acquisition of the data from an analytical method, then DOE is very useful for greater understanding of the method. But in terms of the use of PAT environment, so you might have, as you progress through later stage process optimization, quite a lot of our process colleagues, DOE has been sat in their environment for a long time, so they're very used to running it. So they would try and characterize a purification step or an upstream or whatever it might be through use of DOE. So PAT can be very, very useful within that scenario. So it's just a slightly different slant because you can acquire so much more data so much more rapidly than, say, taking a single grab sample and running it and having that as your value of performance. You might be able to acquire 5, 6 data points instead of just the one, which can really help you understand how your DOE for your purification is looking, so you can understand the different manufacturing, like the critical performance parameters of your manufacturing process. I hope that answers the question. If not, please feel free to provide a follow-up question to that.
Thank you. Moving on, our next question is, how is design of experiments different from sampling strategies, or are these the same?
So the way that I'm reading this question is from a method development perspective, so I'm hoping that's how the question was intended. So when we perform method development, we traditionally from the one factor at a time approach, so for instance, if we take like the size exclusion example, we might just choose a start ionic strength of our mobile phase and vary the pH, but have a fixed flow rate, et cetera, right? So we'll then go, okay, so I'm going to take, say, a 0.2 increments of my pH, go this one's best, and then go ahead and change the ionic strength and maybe go from 100 millimolar up to 300 millimolar at that fixed pH, and go, okay, this is now the best ionic strength for that pH, and then change my flow rate maybe to try and improve resolution. However, I think that when we use the DOE, we gain all of this additional information in terms of interaction effects between them, so that we can basically, and there is definitely a secondary interaction between ionic strength and pH, and there's been a number of vendor publications that support this as well, where you can't look at them as inherently separate things. You need to look at them as a combined factor to understand the true performance for this. So I think the DOE really allows us to do this in a very minimal way, but also one of the benefits of a number of DOE softwares is the visualization tools for being able to communicate the statistical variation that we see. So that's kind of how I see it different from sampling strategies. Again, I hope that was the intent of the question. And again, please follow up if you don't think I've answered it sufficiently for your liking.
Great. Our next question is, are the optimization-based DOE methods computationally expensive?
I would say no. It depends, right? So for our organization, we have a particular statistical software that we have site licenses for. So in terms of outlaying for that site license, there is an associated cost from that, and I don't have those kind of numbers to hand, but I know it's taken care of by our finance, and they've done a kind of return on investment analysis to determine whether it was suitable for investment. But for instance, I can log onto it from the web browser on just my generic laptop. I can create a DOE in under 5 minutes, and really it comes down to me executing the DOE as the limiting time. So in terms of computationally expensive, I'm assuming like that is more around, is my computer going to slow down when I run it? No, I can have everything operating in the background. The softwares that are able to do this these days are very intuitive with minimal training. There's a bunch of online stuff to be able to support the use of these as well, where you can almost self-educate, which is how I sort of did it from my background. And then just talk to colleagues. There's an increasing number of these that are being presented at conferences as well in terms of DOE approaches with the analytical quality by design aspect to it as well. So I would recommend reaching out to people through either environments like this or that. And in terms of actual implementation in, say, your working environment, computationally, no, it's not that difficult. It's about communicating the message of the benefit that can be occasionally more challenging because people aren't overly familiar with statistics. So being able to put that in a much more kind of common understanding is exceedingly useful for them.
Well, thank you for that answer. Thank you for your answers, and thank you, audience members, for your questions. Before we finish the webinar, I would like to ask our presenter, Dr. Springall, if you have any closing remarks for our audience.
I would just like to say once again, thank you for the invite to present at this webinar, and thank you everyone who has decided to join. I hope that this has provided a bit of an insight into how within BPD and R&D Pharmaceuticals at AstraZeneca we are utilizing these kind of tools in an analytical sciences environment. This is always an ever-evolving space. So we do try and keep an eye out on new ways of working with this kind of approach in mind. However, one thing I will say is that we have found this to be exceedingly useful, not just for high throughput analytical methods, but for kind of our traditional lot release and stability methods as well. And we have fully implemented this across a variety of technology platforms now for method development and assessment. And I truly believe it saved us an enormous amount of time, and we have a greater method understanding because of it.
Great. And I would like to take this opportunity to thank our presenter once again. That was Jeremy Springall, PhD of Analytical Sciences, R&D, Biopharmaceutical Development at AstraZeneca. Thank you for sharing your knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers sent back to you. Thank you, everyone. Have a great day.